Hello, I'm Dr. P.R. Padma, Professor in the Department of Biochemistry, Biotechnology and Bioinformatics at the Avinashalingam Institute for Home Science and Higher Education for Women, Coimbatore. You have now chosen to do the module on the applications of animal tissue culture in food industry, which is part of the Food Biotechnology paper. The survival and well-being of all life on earth depends largely on adequate nutrition, and man is no exception. Thus, the growth of food, its processing, storage and consumption is very vital and needs meticulous consideration. Food industry is a thriving industry in which all these aspects are taken into consideration. Recognizing the need for feeding more and more human beings with well-balanced and adequate food, nutrition scientists all over the world are constantly trying to devise ways of improving the production and the quality of food. In this attempt, tissue culture has been recognized as a technique that can contribute immensely. This encompasses both plant and animal tissue culture. Both of them have several applications in the food industry. The major applications of plant tissue culture have been dealt with in module 27 of the Food Biotechnology paper. This module deals with the major applications of animal tissue culture or ATC in the food industry. Unlike plant tissue culture, the applications of ATC in food industry are quite limited. Though animal biotechnological approaches have revolutionized food industry, the use of ATC per se is not very widespread. Scientists are making several efforts in this direction, but the number of animal products that have been developed in tissue culture and reaching the food market is very limited. This is mainly due to two reasons. Firstly, it is extremely difficult to regenerate entire tissues, organs and animals in culture. Secondly, while plant cells have the advantage of totipotency in culture, animal cells do not. That is, plant cells can be differentiated into plant organs or shoot system or root system or into the entire plant exclusively in culture just by modulating the levels of plant hormones. Animal cells do not have this property. Some of the animal cells exhibit pluripotency or multipotency, which means they can differentiate into specific groups of cells. But to develop them into whole animals exclusively in culture is not possible. Secondly, the ethical, legal and social issues, what are abbreviated as LC, that govern the manipulations of animals are extremely strict and limit the number of attempts that the scientists make in this direction. In spite of such limitations, many advantages have been made in the field of animal tissue culture. Most of the efforts and applications of ATC are directed towards the health sector and medicine rather than food industry. Several pharmaceuticals, growth factors, clotting factors, hormones and such health related products can be mass produced using animal cells in culture. Additionally, ATC is the core of tissue engineering or regenerative medicine. But the production of food related products have not been achieved using animal cell or tissue culture to a large extent. However, the genetic manipulation of animal cells in culture and the introduction of these cells into intact animals to produce transgenic animals have had a direct influence on food industry with many applications. Several such genetically manipulated animals have been developed and are awaiting the clearance of the FDA or Food and Drug Administration to reach the market. Such genetically manipulated animals are called transgenic animals. The transgenic animals can be produced by the genetic modification of cells that are maintained in culture. Let us now consider some of the major applications in the field for which propagating the animal cells and tissues in culture is very vital. Thus, the applications of transgenic or genetically altered animals in food industry are also the applications of animal tissue culture in food industry. First, let us consider the improvement of the nutritional value of the animal food. Meat and meat products have taken an important position among the animal products used as food. It is thus important to ensure their nutritional value. Animal tissue culture and the genetic alterations of cultured cells, which are later inserted into whole animals and expressed, has resulted in improving the quality and the quantity of animal food. Let us now take up the major applications in this direction, one after the other. The livestock animals provide the meat used for food. It is highly desirous to increase the amount of meat in the animals, which would result in lesser number of animals being slaughtered. This has been achieved by introducing and expressing the genes coding for growth hormone and insulin-like growth factor into the cells of livestock animals. When these manipulated cells are introduced and integrated into animals, the resultant animals grow to larger dimensions and produce meat that is lean with higher protein content. One of the major problems with the production and consumption of meat is the increased content of saturated fat in the meat. This is believed to cause ill effects to the health of humans. Using transgenesis on cultured cells, genes that alter the lipid metabolism have been introduced into animal cells. The genes coding for the hormone leptin or the enzyme fatty acid synthase have been successfully transferred and expressed in the cultured animal cells. These cells are propagated in culture and introduced into animals to produce transgenic animals. Such animals produce meat with improved muscle mass to fat content, thereby making these meat products healthier. A similar attempt, which also targets the fat content of animal meat or beef, is to transfer the genes that influence the lipid profile as well as lipid metabolism into the cells of livestock animals. Such a transfer causes the synthesis of what are called as heart-healthy fatty acids. This approach converts a fat-rich, protein-poor diet or meat to a protein-rich, fat-poor meat, which is much desired in the maintenance of health. The next aspect is increasing the quantity and quality of milk and milk products. Milk and dairy products take a center position in the food consumption patterns in all the countries all over the world. While the meat, pork, beef and chicken are considered as non-vegetarian products, milk obtained from cow, buffalo, goat, sheep and so on is accepted as a vegetarian food. Milk meets the protein and calcium requirements of vegetarians to a large extent. It is also consumed widely by non-vegetarians. It is given in large amounts to infants, children, adolescents and young adults as well as to the elderly. In view of this, several methods of enhancing the milk yield and its components has been tried by scientists. This is also considered a priority in transgenic efforts. The focus of transgenic approaches in improving milk has concentrated on increasing the amount of milk secreted by the animals, enhancing the nutritional value of the milk secreted, and secreting pharmaceutical or nutraceutical components in the milk. Among these three applications, we shall consider increasing the amount of milk secreted by the animals first. It is highly desirous to increase the amount of milk secreted by a lactating animal, as the milk caters to its own offspring as well as the humans consuming it. In tune with this, genetic transformation of cattle, sheep and goats have been successfully conducted at the laboratory level for improving the quantity of milk secreted. This is known to have a profound effect on the weaning weight of the young ones and improved growth of the offspring, resulting in shortening the duration of utilizing them for food. Additionally, the composition and quality of milk from the transgenic animals have been found to be the same as the animals without genetic modifications. only the amount of milk secreted is increased, thus providing many health benefits to both animals and humans. The second approach is enhancing the nutritional value of the secreted milk. Milk provides good nutritional quality proteins, fat, and the milk sugar lactose. Experimental genetic transformation has achieved an increase in the amount of all these components in the milk of animal. The modifications have been aimed at increasing specific types of casein in milk. Such casein-rich milk is projected to be very useful in the manufacture of high quality yogurt and cheese. Another approach to increasing specific types of casein like beta casein and kappa casein in bovine milk has been tried. Increased amount of glycosylated kappa casein increases the solubility of casein in milk, which decreases the whey expulsion during coagulation for the manufacture of cheese as well as thick curds. It is also expected to result in better tasting low-fat cheese. These approaches, combined with an increase in the quantum of milk, would be highly desirous in the food industry. Scientists are now attempting technologies to improve the specific milk components and also to increase the amount of milk secreted in the same animal. However, all these attempts are still in the experimental stage and have not been approved for general use and market as yet. Now, let us consider the attempts of secreting pharmaceutical or nutraceutical components into milk. Lactose intolerance is a condition where individuals are unable to digest the milk sugar lactose. This is caused by the deficiency of an enzyme that can cleave lactose into its component sugars, namely glucose and galactose. Individuals who are lactose intolerant have problems digesting milk and need to take lactose cleaving enzymes as drugs throughout their lives. Today, the number of cases of lactose intolerance is on the rise. It is also observed that frequent travelers across the globe also developed reversible lactose intolerance, which affects them during travel to specific geographical locations. This condition improves after their return to their original location. Thus, lactose intolerance is a condition that needs serious attention. A solution to this problem has been attempted using transgenesis. The enzyme intestinal cleaving lactase that can cleave lactose has been transferred and expressed in the mammary gland of bovines. This has resulted in the reduction of lactose in the milk produced by the transgenic animal, benefiting the lactose intolerant subjects. Apart from this, other therapeutic products have also been secreted into milk by genetic engineering. These include insulin-like growth factor or IGF-I, epidermal growth factor EGF, transforming growth factor beta or TGF-beta, and lactoferrin, which improves the growth, health status and the survival rate of the offspring as well as the humans who consume the milk. Many antimicrobial compounds and peptides like alpha-lactalbumin, lysozyme and lysostaffin, which confer resistance against a variety of bacterial diseases, resulting in better animal and human health, have also been secreted into milk from transgenic animals. Apart from these, milk from transgenic animals has also served as sources of antibiotics against several diseases like mastitis in cattle, sheep, goats and pigs, and antibodies that prevent many domestic animal or human diseases. Even though all these transgenic attempts have been successful in the laboratory and have also shown beneficial effects to humans in controlled trials, none of these animals or their transgenic products have been cleared by the regulatory agencies for commercial production or free human consumption. This is because of the ethical concerns of genetic modifications of animals as well as the concern about the long-term effects of these products. The arguments and objections put forth by various organizations which oppose genetic manipulations as a whole are many, and have been dealt with in detail in a separate module of the Food Biotechnology paper in this series. Apart from milk, the antibiotics and other therapeutic proteins like anti-snake venom have also been secreted into the egg white by genetic engineering of the poultry. The secretion of such therapeutics into egg white has also been achieved by non-transgenic approaches and have had limited success. In recent years, attempts to secrete the therapeutic proteins into egg white using transgenesis has attracted the attention of scientists. The next application that we shall consider is achieving increased growth rate in livestock and farm animals. One of the widely attempted and successful strategy in animal tissue culture, followed by genetic transformation of whole animals, is to introduce the growth hormone gene into a variety of animals, resulting in increased growth potential as well as meat production. Such an introduction has been successfully done in cattle, pigs and fish. This approach has yielded good result with the Atlantic salmon. This variety of fish is widely consumed in various forms like smoked salmon, salted salmon, etc. In using animal tissue culture techniques and genetic transfer techniques, the gene coding for growth hormone from another fish species was introduced into the salmon. This transgenesis has resulted in a reduction of the growth time of the salmon by half, and the implications of this observation are enormous, as this would mean rearing more salmon with less geographical area in lesser time and a tremendous increase in the yield of the salmon. In addition to this, there is a significant increase in the food consumption rate of these animals, that is, the achievement of enhanced growth with lesser food consumption. This also has a profound influence on the environmental impact of such food-feed ratio. The Atlantic salmon is the only transgenic animal that has been cleared by the regulatory bodies for commercial production and consumption. Another major application to be considered is the rearing of disease-free animals. Meat, beef, pork, chicken and eggs are food ingredients that are derived from animal sources. The safety of these products depends largely on the health status of the animals from which they are obtained. It is very important that these source animals are free of diseases and deficiencies, because foodborne diseases are a very serious concern all over the globe. In this context, it is highly desirous to equip the livestock animals and poultry with the ability to resist pathogenic attacks. To achieve this, farmers who rear such animals usually administer high doses of antibiotics and other drugs. While this controls the microbial attack on the animals, the high dose of drugs can be present as residues in the tissues as well as in products like milk and eggs, which are obtained from such animals. This can result in inadvertent ingestion of drugs and in worse scenarios lead to the development of resistance to the drugs in the humans consuming them. Therefore, it is very essential that while we ensure that the animals are disease free, they should also be free of drug residues. This has been achieved by transgenesis of animal cells. Genes that confer resistance to pathogenic attack have been transferred into farm animals under laboratory conditions. This has provided resistance of these animals to diseases and has also reduced their dependence on dietary supplements of health mixes and other factors. The approaches in this regard include the development of poultry and livestock resistant to pathogenic organisms like Escherichia coli, Campylobacter, Clostridium species and Streptococcus. Genetic engineering approaches have also resulted in eliminating the susceptibility of the animals to these diseases and the threat that they pose to human health. The best example in this regard is bovine spongiform encephalopathy or mad cow disease or mastitis, which is the inflammation of the mammary gland, reducing the quality and quantity of milk. Apart from what we have discussed till now, there are some other applications of ATC and transgenic animal. As we have already considered earlier in this module, all the attempts of using animal tissue culture and their genetic manipulation to produce transgenic animals have largely remained in the experimental level. In fact, the only successful transgenic animal that has been cleared for commercial production is the Atlantic salmon with increased growth capacity. However, other efforts to develop transgenic animals to achieve desired traits are ongoing in several laboratories in many countries. Some of the major applications in this area include reducing the adverse impact of animal excreta on the environment and improving the efficiency of animal feed and dealing with the decomposition of the carcass of animals used in food industry. The former application is achieved by conferring the animals with the ability to digest phytic acid, thereby reducing the load of phytate in the excreta. This has an enormous advantageous implication on the environment. It also makes possible the utilization of phosphorus in a more digestible form. Another application that is being attempted is the improvement of animal feed by transferring genes to produce tender meat, which is a highly desirable trait. Similarly, knocking out the genes coding for the production of a protein called myostatin also improves the quality of lean meat from animals and also causes quick degradation of the animal carcass, causing it to degrade quickly without a huge load on the environment. Animal tissue culture has been highly successful and is widely used in medicine and pharmaceutical industry. Many products have been developed and marketed successfully using animal tissue culture, which have helped in the pharmaceutical industry. Animal tissue culture is also at the heart of regenerative medicine, which involves growing new organs for transplant of damaged ones from one's own tissue. However, in the food industry, in spite of the success stories at the laboratory level, most of these attempts have not been developed into commercial products as yet. This is possibly because of several bottlenecks in these products. The major limitations in this regard can be discussed as under. The animal systems are much more complicated than the plant systems, which makes the introduction and expression of new genes very difficult. Secondly, in plants, most of the cells express totipotency throughout the life, making them easily amenable to transgenic manipulation and expression. But totipotency is a very rare phenomenon in the animals, observable only in certain specific cells in the very early embryonic stage. This makes the development of whole animals in culture extremely difficult and a near impossible feat, which has not been achieved so far. The animal systems, especially the terrestrial animals, are governed by very strict ethical guidelines, limiting the possibilities of experimentation and implementation to a large extent. Additionally, there are several organizations like the Society for the Prevention of Cruelty to Animals that oppose the genetic manipulation and experimentation in animals, which also influence the number of attempts to make transgenic animals. In spite of such limitations, scientists all over the world are working towards the development of products from animal tissue culture and animal transgenesis. It is hoped that animal tissue culture will also provide products for the food industry as it does for the health sector and medicine in the near future. Thus, as we have considered in detail in this module, the possible applications of animal cells that have been genetically modified in culture are many. However, due to several constraints, many applications are still in the laboratory stage only. But the clearance and the commercial production of the Atlantic salmon provides hope that many more such animals may reach the food market in the near future. With that, this is Dr. Padma signing off, wishing you all a very happy learning experience. Thank you.